Okay, let's get started. First of all, with exam one, you all did really well on it. The average was actually quite a bit higher than it typically is. So typically it's right around 82 to 84%. And you guys were, I think the last time I checked, up over 88%. So you did, of those that actually took the test, there's a few people who are still enrolled in the course but don't seem to be doing much. So of those that actually took the test, the average was right around 88% the last I checked. So really good job on the first exam. Having said that, exam two opens tonight. It will go for 1 week before it closes. Exam two will cover from population growth to this first it's going to cover meiosis of what we're going to cover to today. So for population growth through meiosis, I had to move population growth onto exam two, yeah. So through meiosis today is where we're going to cover. So you'll want to have natural selection in there. You'll want to have population growth, genetics, Hardy-Weinberg equations. All of those will be covered on this. So natural selection postulates, Hardy-Weinberg assumptions, which we'll go over today. Plenty of assignments and just that tiny bit of genetics that we've talked about, plus meiosis. So if you have particular questions about that, feel free to email me or ask me at the end here or at the end of our little meeting. So let's start with Hardy-Weinberg assumptions. The lesson was designed to show you a simulation that we would have done in class and kind of take you through that so we could look at what are all the ways. If you recall from our last lesson, we came up with those equations for Hardy-Weinberg, and the whole point of those equations is so that we can quantitatively see if we have had a change in allele frequency. And the assumption of Hardy-Weinberg is there is no change in allele frequency, which is the equivalent of saying there is no evolution, right? There's no change happening in this particular, for this particular gene. So the Hardy-Weinberg assumptions then are the things that we assume are not happening in order for there to be no evolution to occur, okay? So we focused primarily on natural selection, okay? And that's the first one listed here, and is there's no natural selection occurring. Okay, all of those things that can cause a population to shift, all the interactions with environments and other species that could cause natural selection, we're assuming that none of those things are happening. Okay, there is no difference in who survives and reproduces based on their interaction with the environment. So no natural selection. For Hardy-Weinberg to be in equilibrium, there cannot be natural selection, because natural selection would shift the allele frequency one way or the other, or split it. So we talked about different selective or types of selection. But the point I'm getting at here is that we can't have it if we're going to stay in Hardy-Weinberg equilibrium, if the allele frequency is going to stay the same over and over. The next thing that cannot be occurring is we cannot have gene flow. So we cannot have large, and that's a key here, is large. We cannot have large inflows into our population that we're studying of migrants, and we cannot have large groups leaving our population. So we cannot have large amounts of migration occurring. We can have no gene flow. So you imagine if we had a population that was already large and robust. We had 1 million individuals in this population that we're looking at. But then we have 500,000 new individuals move in, right? And they move in from an area that faced different natural selection pressures over millions of years. So they have, a 0.5 allele frequency. Our population has a 0.8 for P, and this new population of 500,000 individuals, almost a large increase, right? They all move in to this area that we've been studying. Well, that's going to change the allele frequency, right? It's no longer going to be 0.8. It's going to be reflective of that combination of 0.8 and 0.5. And so when we have migration like that, and then the individuals start reproducing amongst themselves with that group that moved in, we're going to have a major shift in our allele frequency, and we're not going to be able to say that we're in Hardy-Weinberg equilibrium anymore. So gene flow is one of the ways that evolution can occur, right? Evolution in the definition that we have of a change in allele frequency over time. So we can have no gene flow if we're going to be in Hardy-Weinberg equilibrium. Gene flow can result in change, and that change over time would be evolution. Next is genetic drift. And this is actually probably the second most common cause of evolution that we found after natural selection. So genetic drift occurs when we have small population sizes. And this would be, for example, let's say we had a group of individuals in the Middle East someplace, and they have an allele that is dominant, and it gives the individuals extreme muscles, right? Just inherently, they produce larger muscles. And so we have this population, right? Well, this allele in our Middle East population is extremely rare. We'll say 0.1% of the population has it. Almost nobody has it. Everyone else is basically has the regular allele for large or for muscle size, right? And muscles aren't controlled by a single gene, but we're just using it for an example here, okay? So let's say, because it's inherited, right? We're gonna say that the population that has this extreme allele frequency, they're all closely related to each other, right? They've passed it from parent to child, and so they're all closely related to each other. And let's say that one of the parents says, you know what, we need to leave. I've had this idea. We have to get out of here, and so they take their small family, maybe a group of 15 individuals. Maybe they get some neighbors or some other people that are maybe frightened into coming with them. They end up with maybe 30 to 50 individuals, right? And they leave. Well, what was 0.1% when we considered the whole population for that musculature allele? Maybe it's something like 15% with this group that just left, okay? Because we took a sample essentially by having those small groups or small group of individuals leave, 30 to 50, right? Because they left, we were essentially taking a sample of the whole population when we pull them out. And it just so happens that they have this one weird allele. And so when we pull them out, we're not actually getting a fair sample of how rare that allele was. We're getting a higher concentration of that group, of that particular allele. And so when they leave and go off and they found a new civilization, they find a new land, maybe they sail across an ocean, found a new land, they grow their population nice and big. It's that new population, it's no longer going to reflect the 0.1% that rare allele was in their Middle East location, but now it's 15% of the population, right? Much more frequent. This is called founder effect. So this is like a subcategory of genetic drift. Genetic drift is when we have a small population size that becomes a new population in a variety of ways, but becomes a new population, and it doesn't reflect the allele frequency of what it left behind, okay? So the allele frequency was 0.1%, but this little group that left, now it's 15%. That's because just by random chance, we didn't get a representative sample of the big group, okay? And so we end up with what's called founder effect, okay? When a group leaves and founds a new population, if it's small to begin with, 30 to 50 individuals, there's a chance, just by chance, we will get an allele frequency that is more frequent in that 30 to 50 individuals than they were in the source population. And so that's genetic drift in terms of founder effect. So now we have our 15% with the extreme muscles in these individuals. They found a new colony, they grow to a large population size, but then maybe, centuries later, natural disasters hit. Hurricanes, fires, earthquakes, just terrible natural disasters all occur. And it just so happens that the population shrinks in size again. In this case, it's because a lot of individuals die off. And they don't survive because of the extreme musculature. It just so happens that the people who had this village that survives, well, a lot of the other people die. This village that survives extreme musculature is actually half the population has it. So now we've gone from 15% to 50% have this extreme musculature. And it was just by chance that they survived, right? The muscles will not help you survive a volcano or fire, right? And so they end up surviving. But now our population has shifted again, right? This one small village survives, and they just so happen to be at 50% for this extreme muscle allele. They survive. Maybe there's only 50 of them again. Maybe there's even 1,000 wouldn't be too big of a population size that you couldn't have some founder effect or some, genetic drift. But we'll say only 50 or so survived again. Our population, everyone else died, and so there's only this small group left behind. And that's once again genetic drift. But this time, it's not because they founded a new population that they don't reflect the 15% that they came from. It's because everyone else died, and they just randomly survived, and they randomly had, because it's a small group that we're selecting from, they randomly had 50% of the individuals had this particular allele. And so what happens is we've shifted. We call that bottleneck. Okay, this is when only a small group survives from our source population. So there's a bit of a distinction between bottleneck and founder effect, but the process is the same, right? We're taking a small sample size of the source population, and it just so happens to randomly not reflect the source. Okay, so genetic drift, bottleneck, and founder effect, two subcategories of that. The key distinction for genetic drift is we have to have a small population size that's coming out of it. Okay, and and hopefully I was blatant enough in my little comparison there, right? That genetic drift is something that can cause genetics to not be what we expect them to be. And so if you happen to go to the church website and go to this particular page on there, it's called Gospel Topics Essay, I believe, something like that. Essays. One of the essays is about DNA versus the Book of Mormon, and DNA and the Book of Mormon is probably the better way of saying it. There are some individuals out there who claim, based on genetic analysis, that they can rule out the Book of Mormon story. And in there, the professors, I've met a number of them that were involved in writing this particular article for the church. They actually refer to genetic drift, because genetic drift shows us, based on the history that's proposed in the Book of Mormon, we would not expect the population that is here today that we could take genetic samples from to match the source population that the Book of Mormon peoples came from, according to the story. Because we would expect, based on the story that's in the Book of Mormon, that genetic drift would have been occurring in a number of different places, right? We would have seen founder effect and bottleneck. Not to mention the source population that we're comparing it to. The Jewish people have experienced a number of migrations and changes and things like that themselves. And so I'm not saying this to say, hey, this clearly shows the Book of Mormon is right either, right? But we should be careful when we have sound scientific reasons to expect the genetic markers to be different. We should question those that want to say, hey, I know how to prove the Book of Mormon wrong with this genetic analysis, because we know there's fundamental flaws with the claim that they're making. Likewise, if someone finds a genetic link between people of Jewish ancestry and people who are native to the Americas, we would not really accept that as definitive proof of the Book of Mormon's existence either, or the Book of Mormon's historical accuracy. And that is because there's reasons why, related to gene flow, that we might see some of those things, right? These are not clean samples that we are taking where migration has been limited. We're looking at tremendous amounts of movement and trying to use genetic analysis that just doesn't work out very nicely. We can't control enough to make sure that our analysis are sound. And so I would just say, to kind of end this little sidetrack here, is that we should be really careful when anytime anyone is trying to make these claims for or against the historical accuracy of the Book of Mormon using DNA, because there are good reasons that we understand why there might be the results that we're seeing. Okay, and we can't, anyone that's denying the confounding variables that are involved here are probably pushing an agenda more than they're just trying to understand what the evidence is. And so both for and against, you got to be careful when people are trying to push their narrative. Just understand that there are limitations to what we can conclusively say with the genetic analysis, and a lot of that is due to genetic drift. So there, random side note for you. Not really random, I guess. It kind of makes sense with what we're talking about. All right. The next one there is we have to have random mating. By that, I mean we can't have certain features in the population that lead to whichever sex is choosier, male or female. We can't have them selecting a mate due to that feature, right? Because what's going to happen is whatever feature they're favoring character in their mate selection is going to become more frequent in the next population because that individual gets to mate more. And so we have to have random mating for Hardy-Weinberg equilibrium to be met. So for example, if we had a population of birds and the females were favoring those with longer tail feathers over those with shorter tail feathers, well, the next population or the next generation would have more alleles that led to that longer tail, right? Because they would be reproducing at a higher rate. And so that is why non-random mating can cause evolution to occur. You'll sometimes see it referred to as sexual selection. And particularly in birds, we've seen a very very extreme examples of this, right? Where male birds will have very ornate things that the females select for in their mate choice, and those things are not always correlated with being good at surviving. Okay, and as a result, you'll if you've ever seen like a ring-necked pheasant, for example, very bright colors, very ornate patterns, and then the female pheasant is designed for camouflage, right? Browns and blacks and break patterns to break up their shape to a predator. So these are very the same species, but sexually dimorphic is what we call it, and that's due to sexual selection. The last one is that no mutations are happening. If we have mutations happening, we can essentially take our alleles and change them to something else, and that change might result in this this shift in allele frequency becoming apparent when we go back to sample our population again. And so mutations are a way that we can have a shift in the allele frequency, and therefore we should assume no mutations to reach Hardy-Weinberg equilibrium. So these 5 assumptions all have to be met for no evolution to occur. Now, just like with the natural selection postulates, you're going to be given scenarios. You'll need to be able to say, okay, based on this scenario, which of these assumptions of Hardy-Weinberg were violated and therefore resulted in evolution occurring. And so you'll have to be able to pick those out. All right. So that's a good question. It does, in terms of practical application, right? Our earlobe shape in humans, right? Whether you're attached or unattached in your earlobe shape, natural selection is not acting upon that. That we do have migration in and out, but it's fairly homogeneous, that particular allele frequency for that particular trait. Certainly sexual selection is not occurring based on that. And while there are perhaps mutations that are related to that, they're not changing the phenotype all that much. And so I would say, strictly speaking, no, Hardy-Weinberg equilibrium isn't really met all that often, if at all. But in terms of practicality, like where do we see big shifts happening? There are lots of traits that we don't see big shifts in Hardy. We don't see big shifts in allele frequency occurring. So it is basically a hypothetical, like you're saying there. But there are some of these are so weakly acting upon traits that for practical purposes, it actually does work. So if you were monitoring it closely enough, you could probably pick up changes in allele frequency related to earlobe shape. But practically, it's basically not evolving for that particular trait. So hopefully that made sense. All right, good question. Okay, let's go ahead and jump to our genetics 1. So the big thing with genetics 1 is to understand meiosis, okay? Essentially, and it kind of introduces our lesson, our scenario here. We want to find out if Jack and Jill are half siblings or not through that fun story. But the big thing I want you to understand is, how do we take an individual's DNA and combine it with another individual's DNA and produce an offspring that has the same number of chromosomes, okay? So we have 23 pairs of chromosomes, 46 total. Those chromosomes exist in homologous pairs. What that means is that the same genes that are on this chromosome number 1 are on this chromosome number 1. So the gene for earlobe shape would be on both of these. Now, they can have different alleles. So I might have the dominant allele and the recessive allele here. Both are controlling that shape. So the gene is there, but they have differing alleles. So every single cell in your body has one chromosome that you got from your mom and one chromosome that you got from your dad, okay? For each pair. Chromosome 2, same thing, 3, 4, so on. So we know then that some way an individual, in order to make a gamete, a sperm or an egg, has to take the chromosomes they have and put only half of them into that gamete. And so that's what we're going to try and understand with meiosis. So I know I added a video to the bottom of this to help us out. Hopefully that was helpful to you guys, but we'll go through it in this with this image that I have over here. Really quick, what I was just talking about, having two copies of each chromosome type, that's called being diploid, okay? Essentially, we want to take having two copies, that's what we all have, we are all diploid, and we want to make a gamete that is haploid, or only has one copy of each chromosome. And so that's what meiosis is. So there are two cell divisions in meiosis. Now, in this one, we have a chromosome number of 4. There's 4 total chromosomes with 2 homologous pairs. So this is a homologous pair, and this is a homologous pair, okay? This one, this individual will say red, they got that from mom, the blue they got from dad. So chromosome 1, they got from this one from mom, this one from dad. Chromosome 2, this one from dad, this one from mom. In prophase 1, our homologous pairs link up. This is a key part here, okay? They're stuck together with their pair. The nuclear membrane starts to break down, and the cell starts to prepare for division by making some things that I don't care that you know about, but these little centrioles and fibers that are coming out here. What they'll then do, these pairs will line up along what we call the equator of the cell, middle, right? And we'll end up with chromosomes lining up, each one kind of at random, right? At random, one that this individual got from their mom, chromosome number 1, it lined up the left. The one that they got from mom for chromosome 2 lined up on the right. It's random, right? This one could have lined up over here. We could do that for all 23 pairs of human chromosomes. So where they line up is going to be rather significant in how they end up. That's what's happening in metaphase 1, line up with their homologous pair. In anaphase 1, the chromosomes are then pulled to one side of the cell or the other, and then in telophase 1, the cell starts to split into 2. This thing called a cleavage furrow forms here, and then we end up with our chromosomes in split apart, and here they are down here, right? We took these and we put them down here, okay? So now by doing that, we had 2 chromosome 1s, 2 chromosome 1s, and 2 chromosome 2s. When we split them like this, a 1 and a 2 went to this cell, and a 1 and a 2 went to this cell. So now we've gone from being diploid here to being haploid before we start meiosis 2. Now, instead of 2 copies of each chromosome, we now only have 1. Okay? Now, there's another tricky thing that's happened before we begin. Here in interphase, before we enter meiosis 1, this chromosome that's right here gets duplicated. So this might be chromosome 1 here. It gets duplicated, and so it creates what we call a sister chromatid, right? And so instead of just one line of DNA, do you see how we have these 2 lines here? Okay. That DNA replication happens first. So these are sister chromatids. They're still stuck together. So we refer to them as one chromosome, even though there's 2 copies of each gene there. So they're still one chromosome. This is still just a diploid cell before it starts meiosis 1. And even though we have these 2 copies of our chromosomes stuck together here, it's still considered one chromosome because they're still just one unit. They're still stuck together, okay? And so Now we're going to go through meiosis two. In meiosis two, they're not going to line up with their pairs because they don't have a pair. They just have sister chromatid, their copy. Now, these copies are the exact same because they have the exact same alleles. They literally were, this one was made from that one. And so we end up with, these ones have the same alleles as each other. These ones have the same alleles as each other, okay? So meiosis 2, metaphase 2, we're going to split the sister, they're going to move to the equator, and then as we move, they split sister chromatids apart. Once they're apart, now we're going to refer to them as chromosomes. They're each a separate chromosome, okay? So we took sister chromatids and split them apart, and now we have sister chromatids here and here, okay? Do the same thing with these ones. So sister chromatids become chromosomes. Now we end up with 4 total cells because we did 2 divisions, right? We did the first division, we had 2, we split both of those, and we end up with 4. These 4 chromosomes then, 4 gametes all have 1 copy of each chromosome, okay? Chromosome 1, chromosome 2, chromosome 1, chromosome 2. In this one, we got dad's chromosome 1 and mom's chromosome 2, kind of flipped it, did the opposite there, right? But we still ended up with a chromosome 1 and a chromosome 2. And that's important because we wanted to get to being haploid, right? Now, with this gamete that has 1 copy of each chromosome, fuses with another gamete from another individual, that gamete will only have 1 copy of each chromosome. And so when they fuse together, then it gets back up to 2 copies of each chromosome. We get back to being diploid. So we have to go through this process. So we go from being diploid to haploid. So when the gametes fuse to make the new individual, they end up being diploid again. If we didn't do this, if we didn't half our chromosomes like this, we would end up with just generation after generation of increasing chromosome numbers, and that can cause a lot of issues. That's the process of meiosis, okay? Now, I'm going to jump back. Some of you may have noticed in my little image here that there's a chunk of blue in this chromosome and a chunk of red in that chromosome. This is called crossing over or recombination. During prophase 1, these chromosomes that are so genetically similar to each other can actually have little chunks break out and get switched between them. So we end up with this, remember the red came from mom and the blue came from dad. Well, for this little section right here, they switched right there. So part of mom's chromosome ended up in dad's chromosome there, and part of dad's chromosome ended up in mom's chromosome. And then they go through this process, and ultimately the gametes that are produced are completely genetically unique from one another, because this one is dad's chromosome 2 with a little tiny chunk of mom's chromosome 2. This one doesn't have any of mom's chromosome 2. So even though they're really similar, that little section that was switched, that was recombined, this one doesn't have. And then we see with chromosome 1 here, right? Nothing that came from dad here, but this one has a little piece of blue in there. So it's not exactly identical to that one either. And so this process of meiosis and recombination, what it's going to do is create a lot of genetic diversity in the offspring, right? No, unless there's twins, which is another aspect of this that we're not going to focus on here. Unless they're identical twins, every offspring that a set of parents have are going to be genetically unique from each other. And this isn't something that just evolved once. We had convergent evolution happen with this, right? We see sexual reproduction like this in plants, in all sorts of organisms. And we have to ask ourselves, why would that be? And based on our understanding of the evolutionary studies that have been done, it seems having a variety of offspring is very favorable, because it makes sure or it doesn't make sure, but it gives you a better chance of having a combination of your genes that's capable of surviving in a given condition. And so rather than making clones of yourself, it's better to reproduce with someone else and produce offspring that have a lot of variation between them, with the hope that at least some of your offspring will be able to survive. So there's some really cool studies out there with snails that can switch back and forth between being asexual and sexual, and they've actually studied when they do that, and it has a lot to do with when their environment is not predictable, right? The more predictable it is, the more safe it is, the more likely that your offspring are able to survive. Those snails will reproduce asexually, whereas in other conditions where there's more risk involved, they reproduce sexually. So that's where a lot of that kind of goes back to. There's a lot of good research related to that. So that's meiosis and recombination. Are there any questions on that? Another term you'll see is called link. So meiosis and recombination. Any questions on those two? Okay. One more complication to throw in here with meiosis is something called linked genes. So when we have this this chromosome here, chromosome 1 from mom, chromosome 1 from mom doesn't have just one gene on it, right? It could have hundreds, thousands maybe, of genes on this chromosome. Well, what happens though is when they all get pulled to this side of the cell, they're all going to be linked together, right? If I get the gene that mom had for freckles, I'm going to get the gene mom had for I don't know, I'm just making this up, eye color. Those are going to be stuck together. The alleles that they had are stuck on the same chromosome if they were originally there. And so when they get pulled to that side of the cell, they're always going to be together unless crossing over separates them, or recombination. So linked genes are those genes that are stuck on the same chromosome and should be inherited together. So I believe I mentioned those again in a video of a future lesson there. Okay. All right. So there's meiosis. Let's talk about
one quick question.
Yeah, go ahead.
So sorry. Are we going to be going over the meiosis anomalies part of that one assignment?
I wasn't going to spend a ton of time on that, but if you have questions, you can ask.
I just had one question. It had to do with the crossing over and the recombination.
Okay.
It was during the example of the 4 flies that basically there was a linked gene and it had to do with body color and wing shape. Mm-hmm. And I'm pretty sure the way the cross happened, there was supposed to be basically 0.25 of each different type, a fourth.
Yeah.
It was 1 to 1 to 1 to 1.
Yeah.
But it ended up being 2 of them got 900 plus offspring and the other 2 had only 200 or something like that. And I was just struggling to understand why there were only, I don't know, why there were only 200 in those ones. And
so it's because the wing shape and was it body color and wing shape? Is that the two?
I think so. Yeah. It might have been body shape. I don't know.
I think it was gray and black and vestigial and normal, maybe.
Yeah, that sounds right.
So those 2 genes in fruit flies are stuck on the same chromosome, right? Mm-hmm. And so if they were on separate chromosomes, if body color was here and wing shape was here, it would just be a 50-50 shot whether they lined up on the same side during meiosis 1 here, or metaphase 1. But when they're linked, they're essentially always going to be stuck together, right? The dominant allele, in that case, they were stuck together. And the recessive alleles, in that case, they were stuck together, right? And so the dominants would always be inherited together and the recessives would always be inherited together. That's why you had those two big categories that were like 900. But the recessive category, or sorry, but when we those two smaller categories, not recessive categories, sorry, those two smaller categories, the reason why they exist is because sometimes there's crossing over, this recombination that separates two dominants from each other, where this piece that broke off here and went over here, that had, let's say that had the dominant allele for body color on it. Now, and then this one that came back on this side would have the recessive allele for body color. Well, now recessive body color is now linked to dominant wing shape, and dominant body color is linked to recessive wing shape. Okay.
Okay.
And so that's pretty rare that happens. That crossing over would separate those two. And the further apart they are, the more frequent it would occur. The closer they are on the chromosome, the less chance there is that there would be a break that splits them up. Okay. And so that's where that comes from, is that in rare circumstances, crossing over broke the one it was linked to and caused those recombinant groups that were rare.
So those recombinant groups are like the populations that only had, I guess, 200 as opposed to like the 900. Those ones weren't really expected, but due to crossing over, we had a small.
Yeah. Well,
okay.
And by expected, I mean, like because we know they're linked, we would expect them to stay together, but crossover created those two smaller categories.
Gotcha.
Right?
Okay.
Now, if we didn't have linked genes, we would expect that 1-to-1 to 1-to-1 that you were talking about there.
Gotcha.
Yeah.
Hopefully okay. Got it. Thank you so much.
Okay. All right. Let's see. Okay. So I just put onto a Word document, and hopefully you guys can see this. A few examples from Genetics 2 that I wanted to go over, okay? So there's a number of different inheritance patterns. The one you're probably most familiar with is dominant and recessive. In this case, I believe freckles are considered dominant. And so Jack didn't have freckles. They were absent. So we know his genotype must be, homozygous recessive. Irma had freckles, so we know she must have at least one dominant allele, but we know that Irma, that's Jack's mom, must have a recessive allele as well. And we know that because Jack ended up with two recessive alleles. He can only get one from each parent. And so we know that Irma has to have one there. And Donald, again, has freckles, so we give him at least one dominant allele, but Jack got one of his recessive alleles from Irma. He had to get the other one from Donald, and so Donald then would have to be heterozygous as well. So now we kind of know, based on that, looking at the inheritance pattern here between Donald, Irma, and Jack, we know the genotypes of all three. Jill and Daisy both did not have freckles, so they both have to be homozygous recessive. Okay, so there's our pattern. Well, what we want to know is, could Donald have been the father of Jill? And so what we do is we do what's called a monohybrid cross. We're going to put Donald's alleles that he can contribute potentially there at the top. We're going to put Daisy's alleles that she can contribute on the side, and then we're going to combine them. Okay, and this is going to give us all the possible combinations that theoretically Donald and Daisy could produce. Okay, and if we do that cross and we see, all right, Jill isn't a possible combination, we could rule out Donald as Jill's father. But as we do this, we see, well, Jill's homozygous recessive, based on their genotypes, could Donald and Daisy have produced a daughter who's homozygous recessive? Absolutely. In fact, there's a 50-50 chance of it, or 1-to-1. So we can't use freckles to rule that out. The next one is blood type. And I know a lot of you struggle with blood type. So hopefully going over this will help. Blood type introduces a complication in a number of ways. First of all, there's what we call multiple allelism. This is when we have more than just the two alleles. We don't just have dominant and recessive. We have three alleles, the A allele, the B allele, and the O allele. We also have something called codominance here, okay? Codominance means that we have two alleles that if both are present in the genotype, that we get both expressed in the phenotype. Both get produced in the phenotype. And so if you have an individual that's heterozygous for A and B allele, they will end up with A and B antigens sticking off of their red blood cells. So if we jump over here really quick, genetics 2, there's a nice table that kind of demonstrates this, right? If you have type A alleles only, you get the A antigen. If you have type B alleles only, you get the B antigen. If you have type O alleles only, you get no antigens at all. But if you have A and B, both A and B antigens get produced. And so that's the difference between codominance and traditional dominant and recessive, is that they both get expressed. Now, both A and B are dominant to O. So if I have a B and an O allele, I will get the B antigens. If I have A and O alleles, I'll get the A antigens. So just a brief little rundown of that. Now let's see, based on what was in the table, Jack is O. Well, the only way to be O is to get two O alleles because it's recessive, right, to A and B. So we know that Jack had to be homozygous for O, little I. There's lots of ways you can note this, by the way. There's lots of different ways to do genetic notation. This is just the one that I was trained with, so I use the lowercase I to indicate that it's recessive. Because it's codominant, I'm using, the I is there for immunoglobulin, which is referring to the type of antigen there. When I get the A, Irma is type A, I know she must have an A allele. And I just use a superscript to indicate that with that it's an A, but I use the uppercase I to indicate that it's dominant over O, okay? So we know that her phenotype being A, she has to have an A allele, but we know that Jack had to get an O from Irma, and so we know Irma's second allele must be that recessive O allele there, the little I. Donald is B, so we know he has to have a B allele, and then we know that he had to be able to give Jack an O allele, and so we know he's heterozygous for B and O. Jill is O, so we know that she is homozygous recessive, and then Daisy is B, so we know she has a B allele. She had to be able to give Jill an O allele, so she has to be heterozygous for B as well. So let's go ahead and do this cross. So we do our first combination there, and then we do B there, and then there. Okay, so we set up our monohybrid cross between Donald and Daisy. When we do this out, we see all the possible combinations here. And one of those four, so 25% of our possible combinations is going to be homozygous recessive. So once again, we cannot rule Donald out as Jill's father based on that cross. So all we're doing, once again, is we're finding the genotypes of the people involved. We use inheritance to kind of help us out with that. We then take the potential parents, we put one across the top and one on the side, do all the combinations that they could produce, and that would be 1 out of 4 would be homozygous recessive. We could also use the terminology for phenotype, right? So in this case, 1 out of 4 would be the type O blood. 1 out of 4 would be, 3 out of 4 would be type B blood. So you can use that terminology if you prefer. All right. Rh factor is a second antigen that exists. It's a completely different gene, okay? So this is what sometimes kids or students will mess up on, is they'll try and combine ABO and Rh as if it's one single gene. It's not, it's 2 separate genes. Now we can look at them together, and that can be helpful, but we have to do it the right way, okay? So Jack is negative. Negative is recessive to positive for Rh factor. Irma is positive, but she has to be able to give a recessive allele to Jack, so we know she's heterozygous. Same thing with Donald. With Jill, we know she has a positive allele, but we don't know what her second one is because we don't know who her dad is, and we know that Daisy is positive, but we don't, based on what we know, we don't know what the second allele is for them. So we're a little bit limited to what we can say here. We could take these two and we could do them together in what's called a dihybrid cross, okay? So in this case, we would have Donald, who would have this genotype for those 2 traits. Then we'd have Daisy, who would have this genotype. And we don't know her second allele, so that's going to complicate things a little bit. But we need to know how to do dihybrid cross. So I'm going to do this as an example here. Let's actually shift this down. There we go. Okay. So I remember when I was in math in like middle school, I learned about FOIL. Okay. And this was a way to distribute things out in a mathematic formula. And I use it to kind of help me make sure that I do all possible combinations of an individual's alleles when there are 2 genes involved, okay? So we're gonna take F and say that stands for front. We're gonna take the front 2 alleles from Donald and put them in play here, okay? So we have a B, then a positive, right? The front 2 alleles for those 2 genes. So that's the first thing we could get from Donald, a B and a plus. The next thing we can get from Donald is the outside 2, a B and a negative. Then we can get the inside 2, that's an O and a positive. And then the last 2, which is an O and a negative. All right, let's go ahead and separate those out a little bit. Okay. Now we do the same thing with Daisy. The B, the first 2, the B with the positive, the B, sorry about that, the B with the question mark, and the O with the positive, and the O with the question mark. Okay. All right. So this is how we'd set up our dihybrid cross. We go ahead and fill this thing in. I'm not going to take the time to do that, but we'd end up with first possible offspring here would be homozygous for B, homozygous for positive, right? This one would be homozygous for B and a positive and a question mark for the second allele. So we do this dihybrid cross that lets us do these 2 traits at once. And that's how you'd set that problem up. All right. Next, we have colorblind. So colorblind, I think we've shifted into genetics 3 here. Colorblind alleles are sex-linked traits. That means that the gene for colorblindness is on the X chromosome. And that means that individuals that are male only get 1 copy of that gene, whereas females get 2. So as we go through our little setup here, Jack has normal color vision. Well, that means he has the big N here, okay, for normal vision. The second one, though, I put a Y in that place. Now, you could leave it blank. I like to have a Y there just to remind myself, hey, that the reason why this person only has 1 allele is because they're male, and this is sex-linked. Irma has the normal allele, but we don't know what her second allele is. Obviously, being female, she's going to have 2 of those chromosomes, but we don't know what that one is. Donald is colorblind, and that means that he has the recessive allele on his X chromosome, and then, of course, he doesn't have a second allele because the Y chromosome doesn't have it on there, and so we put a Y in that place. Jill is colorblind. She's homozygous recessive. She has to have 2 recessive alleles to end up being colorblind. Because of that, we know Daisy has normal vision, but she has a colorblind daughter. It means she had to be able to give this allele to Jill. And so that is heterozygous for Daisy. Okay, so if we do this cross out between Donald, his possible alleles he can contribute. There we go. And then Daisy, she's heterozygous, so 1 of each. All right, we do this cross. Oh, this one should have been lowercase. There we go. There we go. We do this cross, and we end up with, well, we need it to be a colorblind and girl. And right there, we have that as one of our options. So again, we can't rule out Donald as a potential father. So hopefully that was helpful. I know it took a long time to go through that, but a lot of students really struggle with how to set these problems up. So I wanted to go through a number of examples. So we got examples there of traditional autosomal. Autosomal means not sex related, okay? Traditional autosomal dominant recessive. We got an example of codominance. We got to do a dihybrid cross. And we saw an example of a sex-linked trait. Any questions on these patterns of inheritance? Incomplete dominance is a little bit different than codominance, okay? Rather than having both phenotypes show up, in that case, we're going to get an intermediate phenotype, okay? So we're not going to end up with one or the other. We're going to end up with kind of an in-between. And so incomplete dominance is a little bit different than codominance. Still, we're going to see a monohybrid cross with that, things like that, but it's going to end up with a halfway there phenotype, not a both present phenotype like we see in codominance. All right. Any questions on genetics 2 or 3 at this point? Okay. Meiosis anomalies. I went over the anomalies and how they occur to produce triploid or monoploid individuals, right? How that can happen through that non-disjunction occurring. Essentially, the chromosomes don't separate properly, and as a result, you end up with individuals who have, for one particular chromosome, 3 or 1 instead of the 2 that they should have, and there's complications as a result of that. let's see. And then we kind of talked about recombinant DNA, or sorry, recombinant frequencies. That was the second thing I was going to cover from this. Are there any questions relating to meiosis anomalies, recombinant frequencies, or incomplete dominance that was covered in this lesson? Okay.
Super quick.
Yeah.
Also, just while you're on this, I was just wanting to confirm with the straight hair, curly hair, if it was codominance, would that just be a mixture then of straight and curly hair?
Yeah, one hair would be straight, the next one would be curly.
Okay, sounds good.
Yeah. Yeah. Be an odd look for sure. Yeah. Let's see. Yeah. So unless there's questions, I think we'll just with recombinant frequency, using that to map genes on a chromosome. Remember that the further apart they are, the more frequently recombination occurs. The further apart they have to be on the chromosome, right? And that kind of makes sense, right? The more space there is for a break to happen and that swap to occur, as we were talking about in this, right? The more frequent the further apart they are, the more frequent that can happen. If they're really close together on the chromosome, it takes a very specific break to separate them. And so we can use that recombination frequency we can compare the recombination frequencies to essentially place where we think those genes are located on the chromosome. And so that's what it was one of the things that we talked about there. And you'll at least want to be familiar with that for the genetics packet. Well, in the genetics packet, it will give you a list of frequencies. And essentially, you have to kind of piece it together like a puzzle, looking at, okay, D and E recombine at a frequency of 15%, D and C recombine at a frequency of 30%, right? Well, then I'd want to put E closer to D than C is on this map of my chromosome. And so that's something we're going to want to make sure to understand. So, all right. Quantitative genetics. The biggest thing you're going to need to know going forward is how to read a pedigree chart. That's going to be in the genetics packet as well. The squares in the pedigree charts that I use represent males. The circles represent females. And then filled in shapes mean that they have the disorder or the trait that we're looking for, and not filled in means they don't have it. Okay. And so we can use those patterns, just like we were doing with Jack and Jill. We can use those patterns to estimate to determine the genotypes of the individuals involved. Okay. And so that's going to be really helpful, right? In determining that. So you'll see that on the genetics packet. There'll be things getting in there. Just before we go into the quizzes, I wanted to talk really quickly about the end of that quantitative genetics lesson after the pedigree chart stuff. The pedigree chart stuff's the stuff that you're going to need going forward. But I did want you to understand that most traits are not single gene traits. Most traits are polygenic. There's multiple genes that come together to produce the phenotype that you have for any given trait. And as a result, instead of a categorical way of looking at it, it's better to think of it as a continuum of the phenotype, right? Not categories of phenotype, but some quantity in a continuum. And so while understanding the basics of genetics is best with these relatively rarer traits, once we get into actual looking at most of the traits, we need to use this quantitative approach. And we see an interaction between the genetics and the environment, which is what I finished that lesson off kind of discussing, that how your phenotype isn't simply a matter of your genetics, but an interaction between the genetics and your environment, and all of them have an influence. So hopefully that was, it was meant to be sort of an extension of what we wanted to understand with genetics, right? That there's this whole other complicated area to it beyond the basics that we're looking at for this class. All right. Quiz 14. Question 1. If my hypothetical organism has a diploid number of 8, how many chromosomes would I want the products of meiosis to have? So we want to go from being diploid to haploid, right? So if we have a diploid number of 8, we want to get down to a haploid number of 4. We want to get half the chromosomes there. Which of the following diagrams depicts the way you'd want chromosomes to line up during metaphase 1 of meiosis? So in meiosis 1, metaphase 1, we want to have the chromosomes in their pairs, right? We want the chromosome 1s next to each other, the chromosome 2s next to each other. We don't want them lining up like this. This is how they would line up in metaphase 2, end to end, not with their pair. And the reason why is because their pair gets separated from them in meiosis 1. So we'd want A to be the one we'd choose here. All right. Given that chromosomes duplicate in preparation for meiosis, if I were to count the number of sister chromatids within a cell that was preparing to undergo meiosis, the diploid number is 8, how many sister chromatids would I have? So this is that replication part that I was talking about, right? We have to have the pairs of chromosomes replicated first. And so we want to say, well, how many chromosome 1s how many chromosome 1 sister chromatids do I have? Well, I have 1 sister chromatid and 2 when they replicate. And then that same thing happens to the pair, or its homologous pair. And so we end up with 4 total sister chromatids. And I know this naming is confusing, but I want to go over it so that we can understand where it's coming from. Okay. All right. Considering all you know about the process of meiosis, will the gametes produced be genetically identical, genetically unique? Meiosis is not related to genetics. It absolutely is. It drives it, right? It's genetically unique is the one we're looking for. And what's so cool about what Gregor Mendel discovered, when he came up with his laws of independent assortment and law of segregation, right? He actually was describing these rules for genetics he was coming up with that kind of govern the Punnett squares that we do. Those rules actually were describing how this process works, right? That we had to separate the 2 chromosomes of the pair. That's going to separate 2 alleles that I have, right? Law of segregation. And then independent assortment, well, that genes that are on this chromosome will independently assort from genes on this chromosome. And so he kind of was describing this process without actually even being able to see it, which is really cool. Anyway, quiz 15. A woman exhibits all the symptoms of a sex-linked trait. Which of the following must be true? Well, her mother must have also exhibited symptoms. Well, that's not true. Her mom could have been a carrier. She could have been heterozygous, but she didn't necessarily have to have the disease. She didn't have to have the phenotype that showed. None of her sons will be affected. Well, a woman will only pass on X chromosomes to her sons, right? Her sons then they only get 1 X chromosome. They have to come from mom. And if mom has a sex-linked trait that's recessive, that X that the son gets from mom has to be one of the recessive ones. So in contrast, all of her sons will be affected, right? Because she only has recessive alleles on her 2 X chromosomes, and they will only get their X chromosome from her. So all of her sons will be affected. Her mother's father must have had the trait. Doesn't have to be true. Her mother certainly had to be a carrier, but her mother could have got it from her mother, right? So it doesn't have to be the case. Her mother's father was at least a carrier of the gene. Okay, so this woman has two X chromosomes with this recessive allele. She had to have gotten that from her father. And that means her father had to get it from his mother, because his father got his Y from his father, right? So her father had to get the at least one copy from, or could only get one copy, but got his one copy from his mother, which means his mother had to have at least one copy. So she had to at least be a carrier of the gene. In pea plants, yellow is dominant to green, round is dominant to wrinkled. If I have a plant with the genotype heterozygous for color, or homozygous for shape, homozygous recessive for shape, how many unique gametes can it produce? Well, we're just going to do FOIL here, right? So we could take the dominant Y and put it with a recessive R, first two. We could take the dominant Y or put it with the last R, which is also recessive. Well, those are the same type of gamete, right? A big Y with a little R. So even though we do two little steps to get there, they both produce the same type of gamete. Then we do the inside two, lowercase y or recessive Y with a recessive R. Well, in that case, we've now had a new type of gamete, right? Homozygous recessive for both. Then we do the last two, recessive and recessive. So that's actually going to be the same again. So we have one type of gamete that is dominant for Y, recessive for R, one type of gamete that is recessive for Y and recessive for R. So even though we're going to produce 4 possible gametes, only 2, they're identical in 2 of the cases. And so the unique gametes that can be produced, there's only 2. If I mated a yellow wrinkled pea plant with a green round pea plant and got 50% yellow, 50 yellow round, 50% yellow wrinkled, what is the most likely genotype of each parent? All right, so let's throw this up here really quick. We had, okay, yellow round and yellow wrinkled is what we end up with. The only way that's going to work is if this is the cross we are doing. Okay. Or sorry, that one's going to be this. Okay. Of the options we have there. Okay. So again, we could go through and do FOIL and they would all be for our first offspring this, right? Well, whenever we have repeated gamete types like this, we can actually just get rid of those and reduce it down. So we only actually need one row to show the gametes that this first parent can produce. In this one, we have 2 possible ones that could be produced right there, okay? So when we do that cross out, this is the only one that's going to produce 50% yellow wrinkled, and that's this one, and then 50% yellow round, okay? The other options wouldn't produce that for us. So this one, they would all be round because this parent would all give each one a round allele. In this one, 50% would be yellow, 50% would be green in that particular cross. So of the options we have. All right. And then when we are looking at 2 or more traits, we must consider all possible combinations of the 2 traits that can form in a gamete. Use your understanding of meiosis to explain why. That comes back to this, right? If they're on separate chromosomes, which most genes are on separate chromosomes from one another, except for the linked genes that we talked about earlier. If this is where the gene for pea color is, and this is where the gene for pod shape is, okay, those 2 genes, the alleles, the yellow allele could line up on this side or on this side. It's just random how it lines up. And so based on where it lines up in metaphase 1, we can get different combinations of the gametes. And so that's what we're doing when we're doing that FOIL thing, is we're saying, what are all the possible ways the chromosomes could line up in metaphase 1, right? Okay. Quiz 16. Snapdragons exhibit incomplete dominance for flower color. If I cross a red flower and a white flower, what color are the offspring? Well, if it's incomplete dominance and one allele is red and one allele is white, that means this flower has to be homozygous for red. This one has to be homozygous for white. When I cross them, they're each going to give, this one's going to give a white allele to every offspring. This one's going to give a red allele to every offspring. So if it was traditional dominant recessive and say red was dominant, then they would all be red. But because it's incomplete dominant, we're going to end up with a halfway in between red and white. Yeah. And so they'd all be pink. If we were then to take two pink flowers, reproduce them together, right? Then we'd end up with a one red, two pink, one white pattern. If we're reproducing red and white, we should get all pink. How can a man with normal color vision father a daughter who is red-green colorblind? Remember that colorblindness is sex-linked and recessive. So in this case, we have a daughter who has to be little n for color vision, right? Homozygous recessive. Well, she could have gotten one of those alleles from her mother, but she had to get one from her father too. Her father's X chromosome has to have that recessive allele. So if he has normal color vision, he couldn't have a daughter that ends up red-green colorblind because he would have had to have given his X chromosome, which has the normal color vision allele on it. So he can't would be the answer here. All right, we are going to do a dihybrid cross by crossing two individuals that are heterozygous for two hypothetical traits, A and B. So this is the setup that I was showing you here, right? With the blood type 1, right? Except for we use A and B here, right? So this one would be AA or AB. This one would be AB. This one would be lowercase a, uppercase B. This one would be lowercase a, lowercase b. And then we'd see the same thing down this side, right? We'd have dominant for both. We'd have dominant for A, recessive for B, recessive for A, dominant for B, and recessive for both. And this would be individual would be that, and the other individual would be, or sorry, we wanted that to be heterozygous, I believe. So that needs to be this and this, and then we'd have the same thing here. So this individual could produce these, this individual could produce those, and then we do a Punnett square out with that, right? Where we go through and do each one of those possible offspring combinations that could come from this. So I'm not going to go through and do that whole thing there, but you should have come up with 9 out of 16 would have the dominant phenotype for both traits, or 56.3 or 0.563%. So if you did the cross right and you go back, you double check, you make sure that you did FLOYL right, you make sure that you had the right genotypes to start with, and you see, expected it to be 9 out of 16, but when I did the cross, I see a 3-to-1 ratio, right? Which is way more of the ones that are homozygous dominant for both traits I would expect to see. When you see that, that's a good indication that they're actually found on the same chromosome, that they're linked genes. So in that case, they would be on the same chromosome. So rather than having a 50-50 with how they line up, they would always be stuck together on the same chromosome there. All right. We were on our last one here, quiz 17. Kate has a child with Down syndrome. If Kate and her husband do not have Down syndrome, What is Kate's genotypes? Well, this question's kind of here to help you understand that you need to resist the urge to try and turn everything into one of these simple problems that we were just talking about, okay? Because Down syndrome is not caused by a single gene. Down syndrome is caused by a meiosis anomaly, by having a third copy of chromosome 21. And so this is not, there's genotype doesn't make sense here, right? What we're seeing here is a meiosis anomaly. And so none of the above should be the correct answer there. Blood types exhibit codominance. If I cross an individual who is heterozygous for A and O with an individual who is heterozygous for B and O, what possible blood types can they produce? Well, we could have the A and B end up together. We could have the A end up with the O. We could have the O end up with the B, or we could have the two O's end up together. And so you actually could produce all four types if this were the cross that you were doing. The genes for polka dots and antenna length and horns in Mendelians are linked. Given the recombinant frequency between polka dots and horns is 38%, between polka dots and antenna length is 7%. So we know dots and horns, they're theoretically 38 units apart. And we know that dots and antenna are 7% apart. What we don't know is antenna length towards horns, or is it away from horns, right? So if it was toward horns, we'd expect to see antenna length and horns have a frequency about 31%. But if it's away, we'd expect to see 45%, and that's what we see here. And so that tells us, okay, that means that horns has to be on one side, antenna has to be on the other side, and then dots has to be in the middle. And so that's what we see here, antenna, dots in the middle, and then horns. All right, assume the pedigree chart below is depicting inheritance of a recessive disorder. What would the genotype of the original mother in the chart be? Okay, so it's a recessive disorder. That means she doesn't have it. Okay, her husband does. So he has to be homozygous recessive. We know she has at least one dominant allele, so she has to have at least one A. Well, when we look at the pedigree chart, we see that she had two children that had disorder. Well, dad, who was homozygous recessive, could have given them one allele. The only way they get the second allele is if they get it from mom, and that means that mom has to be heterozygous, big A, little a. All right. Given what you know about his parents' phenotype, so his mother has wavy hair and his father has curly hair. They both have red hair, so we don't need to worry about that part. All right. Let's see. It mentions earlier that curly exhibits incomplete dominant over straight. So that means if we have both alleles present, we end up with wavy. So that tells me his mother must be heterozygous. His father has curly, so he must have homozygous for the curly allele. So if I do that cross out, essentially dad has to be this, mom has to be this, and this is what our Punnett square would look like. And so it certainly is a possibility that Miles could end up, could have had wavy hair. And if he has wavy hair, there's a potential that the children could have non-curly hair. And so Wanda's controlling mother won't allow it. All right, so 50% is the probability for that one. If we look out at the cross there, 2 out of 4 would end up with wavy, or 2 out of 4 would end up curly. Okay. They later find a picture of Miles when he was young and find that he had wavy hair. Her mother forbids them to marry. Do not consider this when calculating answers for question 1 and 2. Wanda's mother, Harriet, dies of Huntington's disease. Huntington's disease is a neurodegenerative autosomal dominant disease that starts showing symptoms in mid-adults. So when like 30s to 40s, as did Harriet's father. So we know Harriet had it and her father had it. However, Harriet's mother did not have the disease. Wanda's father also did not have the disease. So what is the probability that Wanda will also die of Huntington's? Well, Huntington's, you have to have the dominant allele to get. So father doesn't have it. He has to be homozygous recessive. Harriet did have it, but her mother didn't. That tells me that Harriet has to be heterozygous. So we can use this same Punnett square, right? We could just change this to be little h for her father, big H, little h for Harriet. That the big H gives her Huntington's, and we'd still end up with a similar pattern here, right, of 50% here. So 50-50 whether Wanda gets Huntington's disease. All right, Wanda is of course saddened by the loss of her mother, but now she is determined to marry Miles. Wanda begins to read her mother's journals to get to know her better. While reading, she discovers her mother had a sister who died of cystic fibrosis, which is an autosomal recessive disease that frequently causes death due to lung failure. If Harriet was a carrier, what is the probability that Wanda is also a carrier? Wanda's father was not a carrier. So we want to know about Wanda herself. We're assuming that Harriet is a carrier. That's what the question tells us to do. So Harriet's heterozygous. Her father is homozygous dominant. And we want to know what's the probability that Wanda is a carrier, that Wanda is heterozygous. So if we go and do that cross out, her father is not a carrier. Her mother is, according to this scenario. And so not a carrier, not a carrier. Yes, a carrier. Yes, a carrier. So 50% she'll end up being a carrier. All right. Wanda begins to read her mother's journals. We already read that. All right. Given what you know about the probability of Wanda being a carrier, it was 50%. What is the probability they would so we find out that Miles has been tested and found to be a carrier for cystic fibrosis. What is the probability they would produce a child with cystic fibrosis? Let me make sure I read that correctly. Okay, Miles has a brother with CF. Miles has been tested not to be a carrier. Given what you know about the probability of Wanda being a carrier. Yeah, this is all messed up. Okay, that's why I changed this. So depending on how you're looking at it, you could assume that Wanda isn't a carrier, and if that's the case, you would have put 0%. You could assume that Wanda is a carrier. That would give you the idea that it would be 25%, so 0 or 25% would be a potential answer. Really, because we don't know what Wanda is, we should do two Punnett squares. We should do because it's 50-50, it's equal, right? So we could do a Punnett square where Wanda is a carrier and a Punnett square where she is not. And that would give us 8 potential options. And 2 out of those 8 would end up with a child that has cystic fibrosis. And so I would do the problem, it would be 12.5% here. But because of, depending on the assumptions that you would make, 0% or 25% might have been an option that you would choose as well. I went ahead and gave credit for whichever of those 3 that you chose. So that one's just kind of introducing this element of, if we're not sure, we do 2 potential options there. So. All right. Hopefully I have everything done now. Sorry about this kind of a crazy day. Anyway, if you have any questions,
thank you so much.
Please let me know. Thank you.